So we're going to talk about these two visual pathways that we ended up at in the last lecture today. And we're going to talk about the evidence for the division between these two pathways. We're going to focus a bit more on the ventral visual pathway, and then we'll talk a bit about neural decoding methods. That's the agenda. All right? Okay. First, let me just go over some of the concepts you should remember from last time. So these are things that I talked about in the last lecture, that the kind of things I expect you to understand and know. So also brief recap, last time we went from the retina up to primary visual cortex. And just to remind you of a few of the basics, we start with 100 million photoreceptors in the back of each eye in each retina. And we have this massive data compression that happens right within the retina. The three layers of the retina basically squeeze that data down to this kind of nice, efficient coding system in which the cells leaving the retina that project up to the lateral geniculate nucleus. So then in the LGN, they make a synapse, not a whole lot of change in the properties, and then they project up to primary visual cortex. Okay? And in primary visual cortex, that's where you see retinotopic organization, orientation columns, that is patches of cortex organized in these bands that respond to specific orientations of lines. And you see ocular dominance columns, a differential response to input from one eye versus the other eye. Okay, everybody with the program? Good. All right. So then we said, okay, once you get up to cortex here, primary visual cortex, what happens next? And what happens next is all hell breaks loose. You get this massive, complicated wiring diagram with dozens of visual areas and all kinds of complicated stuff. And the massive oversimplification that we will engage in for the next 10, 20 minutes is to think of this big mess as composed of two main visual processing pathways, right? Like everything else in here, this is radically oversimplifying. You can see the oversimplification right in this wiring diagram. It's not like there's no connections across, there's loads, everything's connected to everything else to some degree. But to some reasonable degree, there's really a broad division of processing between what happens when you go down from V1 down the temporal lobe, down here, versus up into the parietal lobe. Okay? And that division is often called the what versus where pathways. That this pathway is engaged in telling you what is out there in the world, like people, buildings, street, cars, desks, water bottles, yeah? Whereas this processing pathway tells you more stuff like where those objects are. And as we'll see in a moment, how you would guide action toward them, which is related to where, but not exactly the same. Okay, so that's the broad strokes idea here. And what's the evidence for this picture? Well, it starts way back in the 70s with lesion experiments in monkeys. And so the basic finding goes like this. You train a monkey to do a very simple task. In this case, you hide food under one or the other of these objects. And the monkey learns over training that the food is hidden under, say, that object. Okay? On successive trials, you may switch the position of these objects, and the monkey learns that wherever it is, that's where the food is. That's the what task, because he has to identify what. That object, not this object. Okay, pretty basic. Monkeys learn to do that task. They also learn to do a different task, the where task, shown over here. In this case, the two locations don't differ from each other in the appearance of the object or the features of that object, but this landmark is placed near one of them. So the where task, in the where task, the monkey learns the food is under the object that's near the landmark, okay? And the landmarks moves from trial to trial. Sometimes it's over here, sometimes it's over there, okay? So does everybody get how here the two different tasks you can train a monkey to do, the what task and the where task? Two different ways to find food? Okay, once you've trained the monkey to do those tasks, these experimenters lesioned the whole bottom of the what pathway, the bottom of the temporal lobe in monkeys, or they lesioned the where pathway, the top of the parietal lobe up there. And the finding is that lesions to the what pathway interfere with this task, finding the food based on which object it's located under. But they do not affect the where task, finding the food under the object next to the landmark, and vice versa. Okay? So monkeys with lesions up here can do this task just fine. They can say, okay, remember the food was under that thing, I'll find that thing. They do that fine. But they're not good at finding the object that's near the landmark. Does that make sense? Okay. So the fact that we have monkeys of two kinds with two different kinds of lesions with opposite patterns of deficit, what's that called? And what's so awesome about a double dissociation? And you see how it's really stronger evidence that it goes in both directions, right? If we just had these monkeys who had the deficit in the what task, not the where task, that would be suggestive. What and where seem like they're different in the brain. But maybe the where task is just harder. Maybe the monkeys just can't do the hard task. It's not that what and where are different, it's they can do easy things, not hard things. But then if we find that other monkeys with different lesions can do the opposite, then we rule out all kinds of alternative accounts. Most obviously, the one task is just harder than the other alternative account. Make sense? So this is another example of the power of double dissociations, another really important concept that you should know. Okay? All right. So that's the original evidence for the division between the what and where pathways. Lesion studies in monkeys done way back in the early 1970s, and then later studies done in the 1980s. Okay? But of course, we care about the human brain. And as I've mentioned, the organization of the human brain is similar in many respects to monkey brains. So we can take some inferences from this, but it's really nice to check. We're not monkeys, let's hope. And in all respects, so we need to know whether this is true in humans as well. And the strongest evidence that something like this same division is also true in humans comes from the study of people with focal brain damage. And in especially one case, patient DF, who's very famous in the neuropsychology literature because of the powerful lessons that the field has derived from the particular deficit that she had. This is a very tragic case. DF was, I think, an English lady who was living in Italy. She used one of the showers that, the gas-powered shower that somehow operated badly, producing carbon monoxide in the shower. It didn't kill her, but the monoxide poisoning differentially affected the bottom parts of her brain, especially her ventral visual pathway. Very tragic, very horrible. She was quite young, I believe, in her early 30s, and has retained a reasonable life afterwards, but not without very serious deficits resulting from this damage in her ventral visual pathway. So what her story is that she cannot identify objects at all from line drawings. She can sometimes identify objects if they're in front of her in the world, and it's worth thinking about what are the different cues you have if there's an object right there, a real object, versus a line drawing, most obviously color, shading, stuff like that. But in any case, she's terrible identifying objects in general and really bad identifying objects from line drawings. She can't copy line drawings well. Let me just show you the picture. So here, for example, are some things that she was shown. Okay, she was shown these pictures, and she was asked to copy them. Okay, so here is her copy of that picture. Not good, right? Here is her copy of that line drawing. Not good. However, you might think from this, her whole visual system is totally blitzed. She has no visual abilities, visual knowledge, visual anything. But when asked to draw an apple from memory, that's her drawing. When asked to draw a book from memory, that's her drawing. What does that tell you? That's pretty crazy, isn't it? What does it mean that she can draw this from memory, but she can't copy this? By the way, she can only draw these from memory if she's allowed to keep her pen on the paper. If you move the paper, she can't resume her drawing. What does this tell you? This is just common sense. We're just thinking here. There's nothing fancy. Somebody comes along and they can't copy this, but they can. But when you say apple, they produce that. What does that tell you? Exactly. You couldn't produce this from the word apple unless you had a memory of what apples look like. So it's like your inventory of shapes, of things you know, it's intact. They're in there. And yet, when she looks at this, she can't make contact with that inventory of shapes. She can't say, that's one of these things that I can draw. Okay? So what that suggests is that her visual processing abilities, her ability to look at this and see the shape, is messed up. Make sense? Well, if you say to her, draw an apple, and she produces this. Yeah. Yeah. Years after her lesions, she remembers what apples look like so that she can draw this. But that's not enough that if you show her this, she can come up with the word apple or the concept apple, because she can't properly see this. When you ask her what she sees, this is her best way of depicting what she sees, and it's not very similar to this. Okay? So just to go through this, oh yes, and after she draws this, a moment later, she can't recognize her own drawing. It's kind of poignant, but also deeply informative. Okay? So this is called object agnosia, and it's a deficit in some aspect of visual shape perception. Okay? I actually haven't given you all the evidence yet. You could just say, well, maybe she's just blind, right? If she's blind, maybe she could close her eyes and draw an apple like this. She's not blind, as you'll see in a second. Okay, but everybody get the basics here? It looks like some problem with shape perception. Not exactly, but it's close. Okay, so now the plot thickens. So here's a cool task. So this is evidence that she's not just blind. Okay, so here in this task, patients and control subjects are shown lines of a given orientation. Okay, so and they're asked to orient a card in the same way as the lines. There's a line out on a screen, and they have a card. They have to orient it like this. Controls do this perfectly here. Okay, this has been rotated. Actually, the lines are shown in all different orientations, and the patient matches it, but you normalize it to show that whatever the orientation is, the control subjects can do this. When asked, here's her matching thing. She can't see this at all. Right? Here's the data I meant to be showing you. This is where she's shown an orientation and asked to indicate what that orientation is. Here's control subjects. They can do it great. Here she is. She's at chance. Okay, so she can't see that orientation. Sorry to totally blow that. Okay? Can't see the orientation when asked to explicitly report it. But now here's the cool thing. If instead of just saying, match the orientation with your hand to what you see, if you say, here's a card, like say this right here, piece of cardboard, and here's a slot. This is an envelope. Post the envelope in the slot. What does DF do? Boom. She does it perfectly. Okay? So what that shows you is that if you just ask her what the orientation is, she can't consciously access it and tell you what that orientation is. But if instead you ask her to act, make a motor action that depends on that orientation, she can do it. Okay? So this is what they mean by the division between perception and visually guided perception and visually guided action. Right? I said before what and where, but now this distinction is morphing a bit. And instead what we have in the ventral visual pathway is conscious visual perception of what the shape is, or in this case, the orientation is. Okay? Compared to acting based on that perceived orientation. Okay? So what used to be called the what-where pathway is now more commonly called the what-how pathway, right? Reporting what orientation versus acting, like visually guided acting on the object. Like how would you engage with that orientation? Make sense? Okay. So that's the distinction between action and perception, or ability to report the orientation. Here's another similar piece of evidence. Okay, let me see if I can get it right this time. Let's actually read the labels here. Okay, so here's let's start with the perceptual task. Patient DF and control subjects are shown a bunch of 2D shapes like this. They're shown pairs of two of them and asked to say, are they same or different? Okay, so you can see the shapes are pretty similar, but if I showed you those two, you would know they were different, or those two, you would know they were different piece of cake, right? DF is very, very bad at that. She's near chance. She can't tell you if those two shapes are same or different, okay? But it's not like her visual system can't do anything with that shape information. In fact, some part of her visual system has some of that shape information. If you ask her, instead of to report, are they same or different, if you ask her to pick it up, you get evidence that actually she can perceive some aspects of the shape. And the way you tell is you put little LEDs on the fingertips. You have the shapes, actually little flat shapes like flat stones. in front of her and ask her to pick it up, and you videotape the trajectory of her grasp motion as she reaches for it, right? And what you see is that there are certain ways that you grasp an object, right? So here is, let's see here. Here is DF's grasping. When you grasp that object, this is a good way to grasp it. That's a good way to grasp it. This is a crappy way to grasp that object, right? So you grab objects at their waists if they have a waist, or at their long axis. You don't grab them at some weird diagonal thing. It just doesn't work very well. It would slip out of your hand, right? Like right in front of her. Yes, I'm sure, I don't know data on this, but I'm sure she could trace it at least laboriously and slowly. Probably not. If you were tracing an object, you would look at it, size it up, and just boom, like that. I'm guessing it'd be much slower for her because she can't really direct all of that. So she'd probably go off the line and correct it and stuff, but I'm sure she could do it somewhat because she's not blind again, right? Yeah. Okay, so the point is that this is like posting the object. This is another form of visually guided action. Picking up an object requires you to get your fingers in the right aperture, and DF does that in sensible ways right here. She does, and I was just reading yesterday a review. This original stuff was reported, I don't know if I have the years here, but 20 years ago or so. And so since then, there's been this massive literature, and like any big idea you put forth in the field, all these people come out and say, that's not right, and they come out with all these counter arguments. And so Mel Goodale and David Milner, the guys who put forth this whole idea that it's really a division between the what and the how pathways, were writing this commentary on all the critiques. And they point out one of the critiques was that after a lot of experience, DF does learn to do the explicit orientation task much better over time, right? And so people are saying, well, what do you mean? She can consciously report the orientation, even if you just ask her to report it, and she's not posting it. But then they point out that if you videotape her doing the task, what you see is her kind of making a little covert motion. So she has learned that her motor system knows about the shape. And if she allows it to do a little bit of a preparatory action, she can watch herself and read off her own motor system, right? So, cognition and perception are complex and interactive. And again, patients with focal brain damage, they're not stupid. They have a focal deficit. But they're smart people who often come up with exotic compensatory strategies. And apparently DF has somewhat learned to report orientations by trying to act toward the shape. Okay, so big point here, both with the case of perceiving the orientation of a line and with the case of perceiving shapes. When asked explicitly, she's pretty bad at it, but when asked to act on it, she's quite okay. Make sense? Everybody see how that's a division between some kind of conscious perception of shapes versus ability to act on those shapes. Okay. All right, but again, DF is a single dissociation, right? And I'm not going to try to make a case that acting is easier than conscious perception, but in any case, it's always nice to have a double dissociation. And it so happens that there are also, just as there are monkeys with the opposite deficits, there are also humans with the opposite deficit. So this is patient RV. So first to remind you, here's a control subject doing the grasping task, and these are their grasp points. And DF, as I mentioned before, does pretty well and has similar grasp points to the control subject. Okay? Patient RV, in contrast, makes, that's a dysfunctional grasp point, right? That's a bad grasp point. It's not going to work very well. You're going to drop that object, right? And so forth there. So patient RV has the opposite pattern of deficit. Her visually guided action, I don't know if it's he or her, but anyway, RV's visually guided action is very poor, and in contrast, their ability to do the same different task on these shapes is quite preserved. Okay, again, the opposite pattern showing stronger evidence for this dissociation. Okay, patient RV's deficit is called optic ataxia. This is a general deficit in visually guided actions. I can see I'm blocking the slide for you guys over there. Okay, so that's called optic ataxia, and it tends to result from parietal damage, just like the monkeys who mess up at the where task after parietal damage. And again, it's the opposite. No difficulty describing or recognizing objects. Her object recognition is fine, but if she has to act towards them, she's not so good at it. Okay, so all of that was kind of whirlwind tour of evidence from both monkey lesion studies and human lesion studies for differential functions of these two broad pathways. Any questions about that before we go on? Okay. There's loads of other evidence. I just didn't want to belabor it. We could spend a whole course on evidence for this division, but from functional imaging and lots of other kinds. But instead, what I want to do is dive down on that ventral visual pathway. I should just note that one of the many things that I'm skipping over in this class is all the evidence for the fine-grained structure up in this dorsal pathway. We will, in a month or two, get back to talking about some parietal functions in guiding visual attention, so we won't ignore it altogether, but I'll just note that there's also lots of fine-grained, lovely work showing that there are distinct regions up here that carry out different things. So just as one example, there are two nearby but distinct regions, both in monkeys and in humans, up in the parietal lobe, one which is involved in reaching for actions. So if I go to pick up this pen, there's a whole trajectory that I go for in reaching for it, okay? And that's a different thing than this grasp point that we were just talking about, picking it up like this, not trying to do some crazy thing like that would lead me to drop it. So there's reaching and grasping. And actually there's two different cortical regions, one which is involved in reaching and the other in grasping. Okay, even though those are both parts of how you pick up an object. What it says is picking up an object has at least these two functionally dissociable things. You can see that with nearby cortical regions. Okay, so I mentioned that just as one of the kinds of things we're skipping over here. Instead, we'll dive down in the ventral pathway and look in some detail at how it's organized.